what determines an extended phenotype: genotype, evolutionary history, or the environment. Across the diversity of life, there's a lot of variation in phenotypes, and we see this with very classical traits like eye and bird plumage. Phenotypes are not just physical traits. Some behaviors lead to extended phenotypes, and an extended phenotype is the phenotypic effect of a gene expressed beyond the individual organism and onto the environment. For example, here we have a beaver, and we can think of the classical physical traits associated with the beaver, the fur, its tail, but it also has an extended phenotype pictured here with the dam. There are many factors that influence the expression of traits. For example, physical environment, genotype, and the evolutionary history. A lot of research has been done on how these three factors impact more classical traits, such as eyes and bird plumage, but we were really interested in looking at how these three factors impact an extended phenotype pictured here by the ant nest. Ant nests are great examples of extended phenotypes, and here you can see a large diversity of different types of ant nests. They also differ in nest structure, particularly between closely related species pictured here on the left, but also between more distinctly related species. To look at the relationship between evolutionary history and the extended phenotype, we compared nest structures of two different harvester ant species, Pogonomyrmex californicus and Veramesser andreae. The main takeaway between these two species is while their ranges do overlap here in Southern California, Pogonomyrmex californicus's range extends to hotter and drier climates. To look at the relationship between genotype and an extended phenotype, we compared ant nests among different colonies of the same species. And lastly, to look at the relationship between environment and the extended phenotype, we looked at temperature and humidity because these two environmental factors influence the foraging and nest behaviors of harvester ants. We thought that they would also potentially impact their nest structure as well.
So with that in mind, we pose our research questions. First, are there consistent differences in nest structures between harvester ant species and between colonies of the same species? And are harvester ant nest structures influenced by temperature and humidity? So to address these questions, we went out and collected ants. I collected Pogonomyrmex californicus workers from the Red Rock Canyon Park, and Eva collected Veramesser andreae workers from the UC Sedgwick Reserve. Both of these sites here in Southern California. And we brought these ants back to the lab and placed groups of 50 in our bucket setup, which consists of a 5-gallon bucket filled with sand, covered in a wax lid with a single hole in the center from which the ants can initiate their nest building. Each bucket setup was supplied with water and sugar water and placed in one of our environmental conditions here for 7 days. So we collected from a total of 8 source colonies, 4 from each of our 2 species, and collected 4 experimental groups from each colony and subjected each to one of our 4 environmental treatments, those being cold and dry, cold and wet, hot and dry, and hot and wet. After the 7 days were up, we evacuated the ants from the nest, poured melted wax down the hole, and excavated the nest from the bucket setup, then represented each nest as a network. We then digitized each nest and network, as you can see here. We then measured the nests in various ways. At the tunnel level, we measured the length of each tunnel segment, as well as the mean circumference of each tunnel segment. And at the chamber level, we measured the circumference of each chamber. At the global scale, we measured several measures for each cast and combined them through a PCA and found that PC1, termed nest size, explains 61.9% of the variance. So we used nest size as one of our 2 global measures, along with the presence or absence of cycles for each nest, you can see this lower network to the left contains a cycle, whereas the network higher up and to the right does not.
Moving on to our results. For our first question, are there consistent differences in nest structures between harvester ant species and between colonies of the same species? These were our results. I'll orient you to the figure. This is specifically looking at tunnel and chamber level measures. So each row is a different measure. The top is tunnel circumference, the middle is tunnel length, and the bottom is chamber circumference. On the left, we're looking at the impact of species on these three measures, and on the right, we're looking at the impact of colony ID on these three measures. And overall, we found that Veramester andrii dug nests with wider tunnels compared to Pogonomyrmex californicus, and you can see that in panel A. And we also found that colony ID had an effect on tunnel circumference in panel B. These figures are very similar, except now we're looking at global measures. So our top row is the number of nests with cycles, and the bottom row is nest size. Similarly, on the left, we have species, and on the right, we have colony ID. Overall, we found that colonies dug significantly different chambers of nests with cycles. So here in panel B, specifically what we found was that some colonies just didn't even build cycles in their nests. And we also found that colony ID significantly affected nest size.
We then examined our second question, are harvester ant nest structures influenced by temperature and humidity? And here you can see a similarly formatted graph, except now in the left column, nests are separated by temperature, and in the right column, nests are separated by humidity. And in terms of the environment's effect on tunnel and chamber level measures, we did not find an impact of temperature or humidity on tunnel length, tunnel circumference, or chamber circumference. And as for environment's impact on global measures, we found that both Veramester andrii and Pogonomyrmex californicus dug more nests with cycles in the hot treatment than in the cold. Overall, we found that the factors we examined differ in their effects on nest structure. While the physical environment impacted nest level measures, we found a stronger influence of genotype and evolutionary history on the structure of our ant nests, in that they impacted measures at the tunnel and chamber level as well as the nest level itself.